Good afternoon, everyone. It's very nice to see so many faces that I've seen over the number of years through my own participation in research ocean drilling. And I'm very happy to be able to stand here today and introduce the Asahiko Taira International Scientific Ocean Drilling Research Prize. And as many of you know, this is, the prize is given in partnership between AGU and JpGU. And it is named after Dr. Asahiko Taira of Jamstec, and many of us already know of him, and I don't think I have to go through his accomplishments. We all are familiar with what he has done. But the prize is given annually, and it's for outstanding and transdisciplinary scientific contributions enabled by ocean drilling. And what you see here, this statue, is the award that the recipient receives. It's given annually, as I mentioned. The eligibility is an active AGU membership is not required, though I'm sure AGU would like to have all the recipients be active AGU members. And it must be an active early career, mid-career scientist within 15 years of receiving their PhD of any discipline. It must be making an impact, or this individual must be making an impact in the field of ocean research drilling. And of course, we've only given, this award is 4 years old. It's been given 4 times in the past, and we know many of these colleagues here. These are some of our outstanding young scientists working in ocean drilling. But the recipient for 2019 is with us today. It's Dr. Beth Orcutt, a senior research scientist at the Bigelow Laboratory for Ocean Science in Booth Bay, Maine. And she has received this award for her extraordinary contributions to the field of deep subsurface microbiology and biogeochemistry through the IODP program. Now, before Beth is invited up to give her presentation, I would like to introduce Karen. Karen, you are the nominator, so please.
And how do I get mine? Yep. You got it? And then close that. Perfect. Okay. So Beth is receiving this award mostly for her work in deep subsea floor basalt. So if you're not familiar with that as a microbial habitat, it is probably because it's a relatively newly discovered one. This is a really nice overview image of what the deep subsurface biosphere can look like, the different options that you have for looking for life. And so deep subsea floor basalts underlie a lot of sediments, and Beth is going to tell you a lot more about them. But she was on one of the first papers that described the biosphere that exists in some of these deep subsea floor basalts. And she did this very early when she was in the lab of Dr. Katrina Edwards. But then she went on to do further work in the Juan de Fuca Ridge and showed that actually these microbial communities that are in the deep subsea floor basalts actually have an impact on the sediments around them. They're not just functioning on their own. They are part of a larger system. And this work has led her also to Leg 336 in North Pond, which is very important to show that there's a lot of oxidants that are coming out of these deep subsea floor basalts and diffusing up into the sediments that overlie them. This really changes how we think about the oxidants that are available in the subsurface. We can no longer think of just depletion of something to breathe. Something comes back up from the basalts underneath. So all this work that she's done with IODP over the years led her to be invited to be a co-chief scientist with Gretchen Frugreen to the Expedition 357 to the Atlantis Massif, which incidentally was the first, I believe, IODP cruise that had gender parity on it. So it's really cool to see such progress in this science. And I am excited to see Beth's talk. So congratulations, Beth.
Well, it's a real honor to be up here and appropriate to be doing a 4 p.m. talk when all of you got to take advantage of the refreshments outside. And so I hope that we can have fun together, exploring the deep biosphere and what we've learned about it over the last 10 years. Obviously, this work has a lot to do with the drilling program. I wouldn't be up here talking if the International Ocean Drilling Program wasn't a big part of the research I'll be talking about. But it's not in a vacuum, so there's a lot of other agencies that have contributed to allowing this work to happen. The NSF, NASA, as well as programs like the Deep Carbon Observatory and CW. And I'd also like to thank the Hansewissenschaftskolleg, which is hosting me currently for a fellowship in Germany, so that I can build stronger relationships with my colleagues in Europe who are involved in ocean drilling. And so I want to talk to you guys today about all the cool things that happen in those veins and the little pockets in the rock, and what we've been learning about and what we still need to discover. And I like to start with the big picture for those of you in the room who don't think about microbes a lot and why we are so fascinated with this idea. So on the left, well, let's start on the right? This planet Earth, the only place in our universe where we know life to exist. And again, you could argue, why is it called planet Earth when it's mostly ocean on the top? But anyway, on the left is a fancy bar chart explaining where we think most carbon in life is on Earth. And so, not surprisingly, a lot of it's in plants, because you see them everywhere. But maybe surprising to some of you is that a lot of the life is actually in microscopic form that you can't easily see with your own eyes, in the form of bacteria and archaea, and even in some viruses. So they make up some pretty significant pie wedges here. But what I like to focus on is the fact that our certainty about how big those reservoirs are could be off by an order of magnitude or so, right? So we really actually don't have a good idea of how much life are in these ecosystems. If you look at those particular groups, and the authors of this paper, the Baron et al. Paper that came out in PNAS last year, right? Not surprisingly, most of the plants are on the surface of land, but surprisingly, the estimation here is that most of the microscopic life on Earth is probably in the deep subsurface. So if we want to understand this major fraction of life on Earth, we need to be looking in the subsurface. And I think that has important implications for how did life evolve on Earth? Why is there all this life down there? And so that's really what drives a lot of the research that I'm involved in, as well as many of the other microbiologists in this room. And so again, for context, for those of you who may not think about these environments, when papers like that refer to the deep subsurface, what is actually in that calculation? So starting on the right, we can think about the continental subsurface and that we access with mines or coring, et cetera. We can also think about marine sediments, which have either come off of land or are dead phytoplankton and other things that sink down to the seafloor. And those estimates that I showed you in the previous slides predominantly come from measurements made in these environments. But there is another environment out in the subsurface, and that's in the form of ocean crust. And when these calculations have been made, they don't typically include any information from this environment because we have so little data. And so what I want to talk to you about in this talk is what we're learning about this subsurface environment and why it's important. And so the motivation to go study this is not just my own, this is part of a larger community, and so I pulled back up this cartoon that was in a Nature feature back in 2009, so 10 years ago, when this CW program was getting kicked off in the US, right? And so this idea that there are many different teams thinking about what is the deep biosphere, how can we study this, and how can we do this in a collaborative way that is larger than the sum of its parts, and using this as a springboard to really focus also on ocean crust. Because again, we had data from near shore, maybe a little bit of data out here, but we didn't have any data from down here. And so the NSF sponsored the CW, which is a science and technology center, starting in 2009, 2010. And this was the kickoff meeting to get this idea going. And I like to bring it up to remind us how young we used to look, but also to remind us of two key players that unfortunately are no longer with us and part of this work, but they are definitely strong drivers for where we are today. And that's Katrina Edwards and Jim Cowan, who kicked off a lot of the work that I've been involved in over the last 10 years. Okay, so here's the rock from the seafloor. Where are the microbes? What are they doing? These are the kinds of questions that I've been interested in. And of course, the way that we get them is by using vessels like the JOIDES Resolution, or seabed rock drills or other ways to get down into the subsea floor and recover samples. But not only that, also to put in observatories into these holes so that we can do manipulation experiments. We can study these systems over long periods of time, as conceptualized with these cartoons that were made on Expedition 327, come back with ROVs, submersibles, et cetera, and try to study these microbes living in this environment. And so what I want to talk to you about today are results coming from 3 different expeditions that I've had the luck to be a part of. So I'll start with this environment off the coast of Washington in Canada, which is a sediment-buried ridge flank system near shore, so it's relatively warm and anoxic compared to a global ocean crust. And that was drilled on Expedition 327, and there's been many subsequent cruises going there. I'll then talk about the North Pond site out on the Mid-Atlantic Ridge, which is more representative of cold and oxic basalt systems. And I'll end with some of the work that we're doing on the Atlantis Massif, which is an ultramafic uplifted mantle type system that was drilled further north on the Mid-Atlantic Ridge. So starting with the Juan de Fuca environment, this is a site that has been part of the ocean drilling program for a long time. There have been many expeditions to this region to try to understand or use it as a type of model system for understanding hydrology in the subsea floor. How is fluid moving through flank systems, both looking at permeability, porosity, et cetera. And starting many years ago, microbiologists started to piggyback on these types of expeditions and try to use these same observatories to also explore what is the nature of the microbial communities that are moving along with those fluids, and how are they interacting with microbes that might be in the rocks themselves. And so in Expedition 327, new holes were drilled along this presumed flow path to do really awesome tracer injection experiments that Andy Fisher and others led. And again, the microbiologists were part of this and wanting to understand the microbial systems by deploying experiments in these boreholes. And so one of the major questions that I had as part of this team was, what are the microbes that are living on rocks? And more specifically, is there an influence on rock type in the microbial community structure that results in these systems? And that work was driven by work that I had been involved in as a postdoc with Katrina, where we had done a survey of all the data we could find about microbial communities living on subsea floor rocks. Not subsea floor, sorry, seafloor rocks, or hydrothermal chimneys, or sediments, and comparing those communities to one another. So what this is a dendrogram looking at community similarity with a variety of different metrics. And the take-home message of that paper that came out in 2013 was that you find similar communities based on the kind of ecosystem that they came from, and that rock microbial communities that lived on inactive sulfides were different than microbial communities that were on active hydrothermal chimneys. And it depended on the type of mineralogy of the chimney, what those communities were. And so I wanted to take that same concept and explore it in the subsea floor. But this can be really difficult with just working on this with cored samples, because the typical way that you do this is you take a rock from the seafloor, you try to bulk extract all the DNA molecules, the nucleic acids, and then you try to sequence the DNA and compare those sequences to all the other sequences in databases to try to figure out, where does my microbial community sit, where are the different microbes, how do they compare to other bacteria, archaea, and other forms of life. This is an older tree, but to give the concept of how this works. But this is problematic with subsea floor rocks because they come up all contaminated with seawater and drilling mud and all kinds of other stuff. And so you have to sometimes use really aggressive methods to remove that contamination. And sometimes you lose the signal of what you're trying to look for. So is there another way that we can try to do this? And that's work that I started when I was a postdoc with Katrina, of coming up with colonization experiments where we could put different types of minerals into the subsurface in these observatories and let the system reset after drilling and see what microbial communities grow on these. And we can put down different types and see, is there a difference in the microbes that grow on a pyrite versus a basalt versus olivine versus whatever else you want to test. And so we did this on Expedition 327 as part of the expedition. After the holes were drilled, new observatories were installed, and we put some of these experiments downhole and left them there for 4 years. we not only put them downhole, but we also put parallel sets of experiments at the sea floor where crustal fluid from down here was pumped up or allowed to passively flow from this system up here, but it cooled down along the way. So we could also look at temperature as an impact on microbial community structure. And so I wanted to highlight again that this is not just me, this is a whole community of scientists that are involved in this work at Expedition 327 in Juan de Fuca. Andy Fisher, Jeff Wheat, and Kier being major players in that, as well as many of the microbiologists and geochemists that have been part of this. And we've done many cruises together to get these samples and come to the conclusions that we've reached recently. And so again, to orient you to what we did in this paper that just came out a few months ago, is we put our experiments at the sea floor and in the subsurface. They're accessing the same crustal fluids, they're just at different temperatures. And so down here, the temperature is about 65 degrees. Up here, it's about 2 to 4. And we let them incubate for several years. And so what you're looking at here is a representation of the different types of microbial groups that we found on our different rock types, organized by the cold ones up here and the warm ones down here, the basalt or pyrite indicating the mineral type. And you notice here I didn't put a legend. I'm not telling you which types of microbes because that's not important for the point I want to make. The point I want to make is that the cold ones are very different than the warm ones. And also that there doesn't seem to be a major difference between whether you have a basalt or a pyrite. You have similar microbes growing on those different substrates. And you have more variability in the warmer ones as well. So if you take this data and you put it into a way to compare community to community to see how similar or different they are and how they group together, what we can see from that analysis is that temperature is a stronger driver than rock type in determining the microbial communities that we get. So for instance, these samples here that are in blue came from the cold ones, whereas if you want to look at mineral type, again, pyrite basalt, they're up here, pyrite basalt, they're here, pyrite basalt, they're here. So those are grouping closer together than they are driven by temperature. So for me, that was interesting, that in the time scale of these experiments over 4 years, the microbial communities that grow on these different mineral types basically look similar to each other if they're at the same temperature, but if they're at different temperatures, you get different communities. Well, that's not like groundbreaking, but I think it's important to think about in terms of how these communities develop. And that isn't to say that if you let these sit down there for decades, you might see something different. And there may be particular microbial groups that vary between these, but at a community level, they're similar. Okay, so if we take that same data from our samples and say, here are the different microbes that we have that grew on our rocks, and we consider those a representation of the biofilm. And we want to say, how does that community compare to what's in the fluids that are moving past those biofilms? And we can do that because our colleagues in Microbe Hayes' lab have been studying the fluid microbial communities of the system for quite a long time. And so we can compare our data set to their data set and say, do we find the same microbes or not? We also have another comparison data set coming from a cool anoxic site, so not from the Juan de Fuca, from this North Pond site that I'll talk about later. And we say, do we find any of those microbes that grew on our biofilms? Do we find them in cold anoxic fluids from a system that's in another ocean? And so what we can see is that for the samples coming from the warm biofilms, that there's some overlap with the microbial communities that are recruited from the crustal fluids, and not so much from the cold ones in that site. But from the biofilms that grew on our cold incubated minerals, there's actually overlap with crustal fluids from this cold anoxic site. So to me, that's awesome, because it shows that there's global distribution of some of these microbial groups, and that if the conditions change in a way that allows those microbes to grow again, they will become more prevalent. And so that's just, how would I put this? In microbial ecology, this has been a long-term discussion about, like, is everything everywhere and the environment selects? And data sets like this now allow us to explore that in different ways, to investigate the connectivity of this global subsurface biosphere. Now, if we take the same data and say, okay, the microbes that recruit to the crustal fluids, we're going to call biofilm microbes, and the other ones are going to be planktonic microbes, can we see, are there functional differences in these communities? And so what you're looking at here is a representation of the relative abundance of different genes related to biofilm formation and different genes related to movement, so flagellar assembly, and their relative abundance in the data coming from our biofilm fraction and from the remnant plankton fraction. And the point I want to emphasize is, yes, there are functional differences between these categories of microbes, and that the biofilm formers have a relatively higher abundance of genes related to biofilm formation, whereas the plankton fraction has a higher relative abundance of genes related to movement. And so the point here is that when we think about how we look at a microbial ecosystem in the crustal subsurface, we can't just look at rocks or just look at fluids. We need to be looking at them both to understand what the function is in these ecosystems. So we don't just do these biofilm experiments in the Juan de Fuca. These observatories are wonderful playgrounds for doing in situ experimentation of all kinds. And so this is a picture of what one of these observatory looks like, the cork. You'll see all this rusty water, or rust behind some blurry water. That's because the hot water, when it comes out, shimmers. And this is an example of how we might, for instance, collect crustal fluid samples, but we can use this same infrastructure to do new experimentation to study what these microbes are doing. And so we've done that also, collecting some of these fluids and using new approaches like single-cell genomics to explore what can these microbes do? Who are they? And the reason that we're so interested in that, in this system in particular, is that there's not a lot of energy available to support microbial life in this ecosystem. So what you're looking at here are calculations of how much energy is available for methanogenesis or sulfate reduction, based on data that Tina and others have generated. I saw Tina walk in here earlier. Measuring hydrogen concentrations, methane concentrations, et cetera, and calculating how much free energy is available. And so you can see that in some cases, the data plot right on the line of 0 kilojoules per mole per electron. Like, there's no energy for this reaction. And in some cases, it's just barely any energy to support reactions. Same for sulfate reduction. It's not a lot of energy available depending on which electron donor you're using, hydrogen, methane, or acetate. Perhaps carbon monoxide is an important electron donor in the system because it actually can potentially give you more energy. But the challenge is we have no idea what the concentration of carbon monoxide is in the system because we hadn't tried to measure it before. And so by doing these thermodynamic calculations can help guide what are the metabolisms that might be occurring in the system that might not just be your standard methane oxidation or methane generation. And so there are microbes in the system. How are they surviving if there's not enough energy for these types of reactions? And so we use approaches like single-cell genomics where we can take these fluids, sort out individual cells, and interrogate their genomes and ask, what can you do? What genes do you have? And so not to belabor this point, but we had a paper come out earlier this year led by Stephanie Carr, who used to be a postdoc in my group, where we found some single cells that were related to a group of archaea called Hydrothermarchaeota, which branch very early on in the archaea, so a deeply branching group. And when we look for various genes related to metabolism, what we found is that indeed this group of organisms seems to have a possibility to use carbon monoxide in their metabolic pathways. Originally, when we first started investigating this microbe, we thought maybe it's a methanogen because it has all other kinds of genes related to those pathways. But on exploring it further, it looks like carbon monoxide is more important. And also that this organism can use sulfate. And so by applying these new approaches, we can get a better idea of what the life is like in the system and how it might be surviving. And so using this information then to guide cultivation experiments to explore if these reactions are actually happening. So we were lucky enough to get funding from the NSF to go back to the site just in May of this past year to do some experiments to investigate these things. So I put up here a picture of Stephanie because she was very excited about cultivating carbon monoxide oxidizers. But we had a whole team of early career scientists part of this work, including postdocs and a technician from my lab, to try to investigate this. And so what does this look like? So here's a little movie. Thank you, Adam Price, for putting those video clips together for us. showing how we do this kind of work. So we go back to these old observatories, and they weren't necessarily designed for this work. So there's a lot of rust and crud and all kinds of other stuff that we got to get out of the way first. Got to open up those holes, keep very good track of what we do so that the folks who need the pressure data know what we did. But you can see that as we pull out the plugs, hot water starts spewing out of this. We basically created our own little hydrothermal vent to do our experiments at. And so we can put in systems that will allow us to do incubation experiments on the seafloor. That was the main purpose of this recent May cruise. It doesn't look very fancy because I like to build my research from Walmart. But the idea was we wanted to do experiments where we could put stable isotopes on the seafloor under high pressure and high temperature and do experiments to track microbial activity in the subsurface. And so each of those little things you see us working with contains syringes preloaded with various stable isotope cocktails, and we could fill them up and leave them on the seafloor for several hours to days, depending on our luck, and then recover them, bring them back to the ship so that we can apply new approaches to study what's happening in these ecosystems. And so what we're trying to do with this is use these flow cytometry techniques to concentrate the cells from these low biomass samples. So every given milliliter or teaspoon of water from this site has maybe 1,000 to 10,000 cells. In comparison, seawater has like 1 million or higher cells in the same volume. And so, for example, what you're looking at here is flow cytometry data comparing what our crustal fluid looks like when we look in the gate for where cells should be versus all kinds of other particle junk, versus a seawater sample run in the same parameters. And the way that these flow cytograms work, this is an estimation of size of the particle on a log scale, and this is an estimation of fluorescence of those particles, again on a log scale. And this is just emphasize to you that we don't have a lot of cells in these samples, but using these techniques, we can concentrate those cells in very small volumes so that we can work with them. And so what we are doing with these techniques, oh, that wasn't where I meant to go. We can now take these samples that have been incubated on the seafloor under in situ conditions with our stable isotopes and be able to bring them into the laboratory and do mass spectrometry, single cell genomics, et cetera, and actually sort them based on activity to figure out what these cells are doing. And so this is work I'll talk about a little bit more in a second. So that's some of the newest, latest, and greatest from Juan de Fuca. I want to switch gears a little bit and talk about the North Pond expedition that happened in 2011. And some of the similar approaches we used at that site, but also some of the different questions we wanted to address there. So again, this is a site we went to with the JOIDES resolution because we wanted to get into seafloor ocean crust that was underneath a package of sediment. And so this was the most appropriate tool to do that. So to orient you, the site we're working at is, again, a site that's been part of the IODP family, the ODP family for a long time. In this site off the western flank of the Mid-Atlantic Ridge, around 22 degrees north. And when you zoom in off of the western flank, there's a little pond of sediment in between higher outcrops of rock. For instance, hole 395A, which has been part of ODP for a long time, is down here. So we went back to the site to try to understand what are the microbes like in ocean crust in this system. Again, I just want to highlight, this is not just me, there are many people involved in this work, and again, Katrina and Jim were major players in getting this expedition going and all of the hardware that we needed to do these expeditions, and I'm very thankful for the guidance that they gave in helping this get going. And so as Karen mentioned at the beginning, one of the roles of my work as part of this expedition was to measure oxygen concentrations in the sediment that overlied basement at these sites. So what you're looking at here are plots of oxygen concentration with depth at the various holes that we drilled, and these gray lines indicate where we hit basement. And again, part of the excitement of this data was a really strong indication that you have oxygen. There must be oxygen in the basement, diffusing upward, confirming that we have oxic conditions in basement. But more importantly, we can then use this data in comparison to other dissolved elements in the fluids to say, is this amount of oxygen what you think would be there if you just had, advection and diffusion of oxygen as water's moving along in ocean crust? Or can we see any signal that there's been oxygen removal as it's been in ocean crust? And so the way that we did that is with a reaction transport model. estimating, so we have our known oxygen concentration in basement based on what we measured in sediment, and if we assume flow trajectories that in bulk go from point A to point B, although we know from hydrogeology experiments that it's not that simple, and if we say, this is what we have, and this is what we can model the concentrations could be, depending on various flow rates and the distances between these two sites, and the difference between what the model suggests and what we measure then would be considered excess oxygen. And so the lack of hitting that line indicates that there's oxygen removal as water is potentially moving from one direction to the other. And then if we make assumptions about how deep the water has moved, what the permeability of the rock is, et cetera, we can come up with what the potential rates of oxygen removal are. So we didn't necessarily say, aha, there's oxygen consumption by microbes in the basement, but we at least give a bracket for what that rate could be. But it doesn't necessarily have to be from microbial activity. It could just be due to other reactions. And again, there's a big assumption here that we know which way the water's moving. And Adam and others in the audience can tell us that that is hard to determine. Part of the importance of data sets like this is that they can go into compilations of data from all over the world that are giving us a different picture of what subsurface ecosystems are like than we had 10 years ago or so. And so I'm showing here a plot from Steve Donadal's recent paper showing an estimation of how much of the subsea floor has sulfate reduction type conditions that are dominant versus how much of the seafloor has oxygen either penetrating most of the way or all of the way through the sediment into basement below. And I hope what you can notice here is that a lot of the seafloor is oxic. And I think when we were starting to do this work decades ago, and those who inspired me to get involved in this field, that wasn't necessarily an idea that we had. And so I think this is really fundamentally changing how we view our Earth and what these systems are like. Okay, so we didn't just focus on measuring oxygen in sediments, we also did these observatory experiments. That was the major focus of this expedition, to put in new observatories like we did at Juan de Fuca to study what are the microbes like in this system. And I want to emphasize here that the corks that are at North Pond actually are different than the ones at Juan de Fuca. We put a lot of effort in designing corks that would not rust in oxic, cold environments because we wanted to study microbes involved in iron oxidation. And so we put in fiberglass casing, we painted steel, we did all kinds of stuff. It was quite an undertaking. And there are pros and cons to doing this, but one of the pros is that that infrastructure allows us to get really good, clean crustal fluids from the system for studying microbial ecology and microbial processes. And so this is a paper that came out last year, led by Suni Shah Walter et al., where they were able to collect fluids from these observatories over time, look at the organic carbon in the system, both the concentrations, the stable isotopes, the radioisotopes, and get an estimation that this subsurface ecosystem may be responsible for removing like 5% of the organic carbon in the ocean. And we would not be able to make measurements like that if we hadn't put in the time and effort to have these observatories. So I'm not saying go put fiberglass corks everywhere because they are expensive, and thank you very much to the Moore Foundation for helping pay for them. But if the purpose is right, I think it's worth having those conversations about the infrastructure because it allows us to uncover information that we did not know. I don't think there was an appreciation for the role that the subsurface plays in carbon cycling in the upper ocean. Okay, so at North Pond, we deployed thousands of colonization experiments, and they were recovered a little over 2 years ago, and I didn't want to put any of that data in here because this talk is being recorded. So maybe come to my talk on Thursday and I'll talk to you about what we're finding. So stay tuned for that. And then finally, to wrap it up in terms of new work that we're uncovering about the subsurface ocean crust, talk a little bit about the expedition 357, which kicked off in 2015, and I was fortunate to lead with Gretchen Fugreen and many other awesome scientists to study the system a little further north on the Mid-Atlantic Ridge. So to orient you, the Atlantis Massif is a representation of an ocean core complex where you have deeper mantle-type rock that has been uplifted to the seafloor due to a variety of different processes that others in this room are way more familiar with than I am as a microbiologist. The Lost City hydrothermal vents, which I'm sure you have seen in scientific documentaries, are hosted on the Atlantis Massif on the southern wall. And so the justification for this expedition was to drill into the southern wall of the Atlantis Massif to understand not only how did this rock get up to the seafloor, what were the processes that allowed for deformation and uplift and alteration, but also what does a microbial community look like in these rocks? Because the microbes that are in the Lost City hydrothermal vent field are very unique, and they are used as an icon for understanding what life might be like on other planets. And so are the microbes in the rocks that host that system also really crazy and weird? Can they live under high pH conditions, et cetera? And so we wanted to drill into this system, but using the JOIDES resolution to do that at that time was not really the best option because we needed to get through, there wasn't a lot of sediment on the surface, and we wanted to get really intact cores. And so we used a seabed drill that was lowered from a traditional research vessel to core into these sites. So again, not just me, many people are part of this work, and so this is the offshore science party. Our purpose was to collect the core and take out the subsamples needed for microbiology that have to be taken right away, but then the majority of the core went back to Bremen. In 2016, we had our onshore sampling party hosted by ESO at the University of Bremen to collect all these samples and investigate what was going on. And so again, our purpose required a different approach than using the JOIDES resolution. So this was the first time in the ocean drilling program that seabed drills were used. This is a picture of the MEBO drill from Bremen. We also used the RD2 from the British Geological Survey. And we didn't just use them as is, we also worked with their engineers to design systems to allow us to collect samples for exploring microbiology. And one of the main things we did was design a system so that we can inject synthetic tracers into the seawater that's being used as a lubricant to circulate the drills. And that allows us to assess the level of contamination of the samples that we recovered. We also designed fluid sampling systems, all kinds of other stuff. And if you're interested in that, check out these two papers. They'll give you lots of those details. And what did we get? So on the left here is a picture of the Atlantis Massif oriented with north going that way, showing you the sites that we drilled along the southern wall. For reference, Lost City is right here where this triangle is. And I picked two representative pictures of the cores that we took for microbiology just to give you an idea of what we drilled through. And that we pushed these seabed drills to the limit. They were drilling from hard rock into baby powder. This is talc, right? So if you can come up with a rock drill that can do that, woo. So we didn't get the penetration depths that we wanted, but we got some amazing recovery of core from this uplifted system. Here's a representation of the types of rocks that we collected. This comes from a paper led by Stefan Rumejan, giving you an idea that we got a lot of serpentinized Harzbergites, we have dunites, we have also basalts in the system, but they're highly altered, very pervasive serpentinization. And our petrologist colleagues are way better than I am, and reading the book of what this information tells us about how we can reconstruct how these samples behaved, and that you have these peridiotites with gabbro bodies, and that as they're uplifted, you have this pervasive serpentinization going on. The microbiology team then wants to be able to look at these same samples and say, what does a microbial community look like? Well, okay, that's going to be challenging. And so we had to come up with new approaches within this mission-specific platform approach, working with rocks and trying to understand what was going on. And so I wanted to show here two key players for helping us do that, and that's Yuki Murona, who was part of the expedition and generated the cell counts from our rock samples, and coming up with techniques that we could use on the ship to try to process these samples without contamination. And also, then many of our scientists went to the Kochi Institute, the Kochi Core Center, to use their sophisticated sampling equipment so that we could remove the exterior layers of our samples without having to burn them, because we had some organic chemists with us who did not want that to happen. And so remove these exterior layers and get at the interior. And it was worth it to do that because these samples have incredibly low cell biomass. And so what you're looking at here is a representation of how many cells there are per cubic centimeter of rock on a log scale. This is 10 cells per cubic centimeter. This gray area is the minimum quantification limit. as determined by Yuki. And so you can see that some of our samples are right in that zone, where you have like 10 to 1,000 or 10,000 cells per cubic centimeter. These are actually lower than what I anticipated we would get, because when we work in other basalt systems, we get more like 10 to the third, 10 to the fourth. And so that already tells us something really interesting about this shallow uplifted mantle, is that perhaps it's not supporting as big of a biosphere as you find in basaltic systems. Why is that? I think that's something that we still need to explore. But what that also means is kind of the standard approaches that deep biosphere scientists use to investigate the microbial communities in these samples are going to be really, really challenging. You're going to have to extract nucleic acids from huge fist-sized pieces of rock to get enough signal, if you can get any at all. And so we wanted to come up with new approaches to try to look at that, and that's something that my group has been working on these last few years since the expedition. And so I want to highlight a postdoc who was in my group, Jackie Gorgial, who's been working with Nicole Poulton, the director of the flow cytometry facility at Bigelow, to develop this technique of liberating cells from these rock samples and being able to use flow cytometry again to identify cell particles and then sort them into very, very small volumes so that we can work with them. And we can either sort them to a bulk scale so that we can do things like nanosims or metagenome construction, but we can also sort individual cells from these rocks at the single cell level and generate their genomes to explore what they do. And so again, I'm not going to give you the data in this talk because it's being recorded, but maybe come to my talk on Thursday. So why are we doing this? What is the purpose of like trying to squeeze blood from a stone, essentially? And for me, part of that motivation is that these systems represent important analogs for looking for life on other planets, especially ocean world planets in our own solar system, not planets, ocean world bodies in our own solar system. Because if we can have life existing on very little energy in our rocky subsurface, we need to understand how to identify that life when there's not very much energy available and there's not very much biomass, to give us good benchmarks for what we need to be looking for on these other worlds. Beyond that, I think there's also an important role that this science is going to play in the coming years, because there's growing interest in using these crustal resources for industry. So for example, the field of deep sea mining is increasing rapidly, and we need to understand what microbial communities look like on rocks on the seafloor and what they do and how they connect to the subsea floor. And so this is a representation of that kind of environment, an ocean crust system on the seafloor, where we discovered new and interesting animal behaviors associated with fluid rock reactions at these sites. We need to understand the diversity of microbial systems and the important functions that they provide in these ecosystems in order to inform that industry. Likewise, there's growing interest in using subsea floor basalts and other types of systems for carbon injection and rapid carbon mineralization as a negative emission technology. And so we need to understand what would that do to the microbial ecosystems and the services that are provided in there. And so I think for me at least, these societal issues are going to be what's driving the research that I'm doing moving forward over the next decade or so. And we absolutely have to have an ocean drilling program in order to explore those things. And so I'm excited about the future of the drilling program and being involved in helping shape that framework for the next 25 years. So with that, I will hope that you have shared my love for rocks, and I will be happy to answer your questions about what we're learning and what we need to do next. Thank you so much.
We have time for questions.
Yeah, questions? Criticisms? If not, maybe, yeah, go ahead. Yes. Yeah, so to repeat the question in case folks couldn't ask, when we looked at the biofilm communities that formed in the Juan de Fuca colonization experiments, maybe I can go back. Close your eyes if you get seasick. Almost there. Okay, nope, not that one. So I think what's driving that is that if you look at the communities that grew on our incubations under warmer temperatures, some of them actually look somewhat similar. They have, they're dominated by different but similar to each other microbial communities. Why that is, we don't really have a good handle on. So for instance, in some cases, there's a lot of firmicutes, which are known to be spore formers. And in other cases, we have organisms that don't seem to be spore formers. And so why is that? It could also be related to the methodology we use in terms of extracting DNA from these very small samples, and that there could be amplification bias that shows up in this, so it's not always a good absolute identification of the relative abundance. So that's probably what's driving those separation on this, why you have two samples up here and the other one's down here, and that's what's driving it. There was another hand somewhere nearby. Yeah, Jens.
This extraction that you showed on the rock, it's a gentle.
Yes, I did say that. I didn't say it, but I'll go back to that. So where's Jackie's slide? Yeah, almost there. Okay, yeah, so Jens rightly noticed this point that I had with a star, that we use gentle extraction. So what that essentially means is we try to use some of these more quantitative extraction techniques that yourself and Yuki and others have been really trying to perfect. The challenge that we found is that those, for instance, acids or detergents or things that you would use to try to liberate the cells would interfere with the downstream genomic processing that we wanted to do. Why that is, we're not sure. You know, if it was carryover, if it degraded the cells, we're not sure. Importantly, so what we know is that we don't get all the cells. We only get a fraction of the cells from the community. But what we can see when we sequence samples from this smaller portion of the community, that it's still representative of the larger community. So we can do a gentle extraction, we can do the bulk, the hard, harsh, quantitative extraction, and if we look at the structure of those communities, they look like we're getting the same microbes, which was important to me. Like, we didn't just get, for instance, gamma proteobacteria or something like that. Yeah, and I'm happy to talk to you more about that if you have further questions. Other questions? Yes, Luke?
So you mentioned that you have lower abundance of cells in the rocks than you expected. Was that maybe because you were controlling more strongly for contamination than you had previously? Would you say that was the same expedition where you were applying more controls on contamination?
Yeah, no, that's a good question. So again, coming back to this plot, when colleagues of mine have, so I haven't, let me emphasize, I didn't do any of this work. These cell counts came from Yuki. On other expeditions like North Pond, not necessarily Juan de Fuca because we don't have as good of data from there, but North Pond, for instance, we also try to handle those samples very cleanly. And if you look at cell counts based on either microscopic cell counts or quantitative PCR-based approaches, either in the fluids or the rocks themselves, we usually get 10 to the third, 10 to the fourth. Looking at it from multiple methods, multiple years, those kinds of things. And so I was anticipating that we might get in that same range for the Atlantis Massif samples, but they're lower. And so is it because a lot of these samples are ultramafics and ultramafics can't support the same biomass? Is it because they used to be really, really deep rocks and they haven't been fully colonized? I think there's a lot of questions that we can still explore here. So it's not necessarily that we did it better here, it's just that I think that the data tells us there's actually something different between the rocks. Any other questions? All right. Well, I'm happy to go talk to you down here because we have some time left, right?
Yeah, yes,
Okay.
yes. But if there's no urgent question at the moment, I would like to thank Beth for this very comprehensive and exciting talk about what has been achieved in our study of the deep subsurface biosphere in the marine system. And it's an amazing thing, is it's not that long that we've been actually looking at this. So I think what has been accomplished in a very short period of time through ocean drilling is amazing. And I'd like to thank Beth for this wonderful talk and congratulate her again on being the 2019 recipient of the Taira Prize. Congratulations, Beth.